A little bit past 4, so I think we'll get started. Welcome to the relatively small numbers here in person. We kind of had no idea how this would go, and you know, it's probably one of these things that just will depend on the day, and that it is nice to at least have some of us here in person. We have a lot, quite a large crowd on Zoom. Just a couple of announcements. We will have a meet and greet after this. So there's actually some food in that in the lobby. You know, those of you that have been here for a while know that we have in the past done that before the lectures, and we're going to try to do that after the lectures this year as an experiment, I guess. If you are taking the class that's associated with this, hopefully you all received my emails. I mentioned that I was going to pass around a sign-up sheet. Instead of that, I've decided to have you just send me an email saying that you were here, and that will that'll prevent us from having to actually pass something around, and it'll work just fine. The only other announcement before I introduce our speaker is that next week we'll have another live lecture. It'll be our own Gabe Bowen, who is among several faculty members up for a 5-year post-tenure review. And so we're having a new tradition of having those of us, including me, but since I'm one of the organizers so far, I've been able to escape this, but we'll see if that happens next semester. But it's my pleasure to introduce Katrina Twing. She is currently an associate professor at Weber State. Katrina is a geomicrobiologist who uses DNA to study the analysis of microbes in extreme environments, and especially in the subsurface. She got her PhD at Michigan State. She did a postdoc actually here at the University of Utah with Bill Brazelton over in biology. She spent some time in Tennessee at Tennessee Tech, and she tells me that she was thrilled to get an offer to come back to Utah, to Weber State. And a lot of her ongoing research here is looking at life in some of the abandoned mines in the Wasatch Mountains. So that'll be something interesting to think about in the future here. But today she's going to talk to us about the microbial ecology of subsurface serpentinite rocks. So Katrina.
Great. Thank you very much, Kip, and thank you all for having me here. It feels a little bit like a homecoming, both that this is the first time I'm talking to a group of people in person in quite a while, and also it's my first time back on campus in a couple of years. So if you don't mind, since we've got some good space and it's a little bit easier to talk, I'm going to go ahead and take off my mask, fully vaxxed. So as Kip said, today we're going to talk about the serpentinite subsurface environment. I recognize that I'm the only microbiologist in here, so we're going to do just a little bit of review of some basic micro, as well as the methods that I'll be using or have been using. Come on. So first, we'll just review kind of subsurface microbiology, talk about some basic microbial methods. We'll talk about serpentinization and what the relevance to life is. And then we'll look at the microbial ecology in serpentinite fluids, and finally in serpentinite rocks. So microbial ecology is really just using, I don't know how to make this thing at the top go away, but microbial ecology is just using basic ecological principles that have been around for a very long time to understand how microbes interact with each other, how they interact with their environment, and probably the most interesting for me, and I would guess you as well, is how they contribute to biogeochemical cycles. Because a lot of times, especially in the last year and a half, we think about how microbes hurt us, or sometimes we think about the microbiome and how they can help us. But the truth is there's a lot of other microbes out there that have a lot of impact on our natural world, not just our health. I really don't know how to get rid of. Okay, well. So the questions that we're asking are, who is there and what are they doing? Those are really the basic questions that are always being asked in microbial ecology. A little bit of perspective on microbes. So again, when we usually think about microbes, people are thinking about how they associate to humans. This is an estimate of the number of microbes in the world associated with animal guts, so our guts as well as other animals. This is an estimate of the number of microbes in the ocean. So you can already see many more microbes in the ocean than associated with animal guts. And here we've got the number of microbes in soils. So that already drastically dwarfs the number of microbes associated with oceans and animals. Of microbial biomass is deep underground. And the truth is, we know very, very little about that. That's concerning. So we know very little about the microbes that are deep underground. And that is a big deal for a couple of reasons. One, like I said, is that they contribute to biogeochemical cycles, and we don't know really what they're doing. And the other thing just has to do with carbon itself, right? We're very concerned about carbon and the carbon cycle, the amount of carbon that's going from the earth up into the atmosphere. But all life, including microbial cells, are made up of carbon. Our carbon budgets of how much carbon there is to cycle through the world really doesn't take into account this vast number of cells that are down there. So we don't have a full handle on the amount of carbon on Earth if we don't take into account these cells. So how do we study subsurface microbes? If you've ever taken a basic micro class, and I fully understand not everyone has, the kind of methods that usually are taught in laboratory classes are culturing. Culturing is just going out to the environment, collecting a sample, and trying to grow the microbes in artificial conditions in the lab. It's what we think of as microbiologists doing. But the truth is, 95 to more accurately, I think 99% of all microbes that are out there in the world haven't been cultured and likely can't be cultured using the methods that we're using. The methods that we use to try to culture bacteria are very human-centric. We're trying to culture microbes in and on our bodies, and it doesn't translate to microbes in other environments. So that's not going to work when we're looking deep underground. Microscopy, so just sitting down, using a microscope and seeing what's there. Using microscopy alone is challenging for subsurface samples, and it doesn't tell you a whole lot. Microscopy alone just tells you the shape of cells or how many cells there are. That can be useful information, but it can't get to who's there. There's about 6 different shapes that cells can take, and there's billions of different types of microbes out there. So just knowing that they're a circle or a line isn't going to help us here. Additionally, I should say that when you're dealing with subsurface samples, you're usually dealing with microbes on rocks, and anyone who has done XRD, XRF, I'm not great with geology methods, but who's been looking at rock slices underneath the microscope knows that you've already got things that you're seeing there. So trying to put microbes on top of that, you get a lot of interference, and it takes a lot of advanced technology to kind of get around that. So what we use to explore these unexplored systems really tends to be DNA analyses. We go out and we get our sample. In this case, this is a rock that I crushed up, or sometimes we collect fluids. So we filter it through a little filter that's about this big. We extract the DNA from those rocks or water samples, and then we sequence that DNA. And just by looking at the A's, T's, C's, and G's, we figure out who's there and what they're doing. This allows us to explore unexplored microbes. Ones that we haven't seen before, we can learn about them. We have to have something that can grow in order to learn about it here. And we either need to link up microscopy with DNA analyses in order to learn about new things using the microscope, or we're limited to really just what the shape of the microbes are. So using DNA sequencing, we kind of have two basic methods, and I promise I won't go in depth on them, but we kind of have two approaches here that we'll talk about today. One is that we can take our DNA sequencing and use all of that computational power and all of the data that we can generate and put that all into a single gene across the entire sample. And so if we do really deep sequencing of just one single gene, what we can do is compare that one gene across all the organisms that are in the sample and all the organisms that we already know of in databases and figure out who's there. Not super relevant for this talk, but the single gene that we use is called the 16S rRNA gene. It's a part of your ribosome. We have them, microbes have them, every living thing has them, which is why we can use it to figure out relationships between organisms and who they are. So one gene can tell us the diversity or who is there. Alternatively, we can take that same amount of sequencing effort that we applied to one gene, looking at it really, really deep, and we can spread that out across all of the genes in the organisms or the sample's really genome. And by sequencing all of the genes in an entire sample, what we can get is a picture of the metabolic potential of the organisms that are in that sample. Essentially, we can get a sense of what the organisms from the sample are capable of doing. So who's there and what can they do? Okay, the part you were waiting for, right? So what I've been studying is microbes in the serpentinite environment. So serpentinization is a geochemical process in which olivine, when it comes into contact with water at the appropriate pressures and temperatures, turns into serpentine. When that happens, it is going to release magnesium as well as hydrogen. And that hydrogen that gets released through Fischer-Tropsch reactions can react with inorganic carbon in the system and form methane and organic molecules. Simply put, we take olivine, combine it with water and some carbon, and then we're going to get serpentine, high pH, energy, and food. So energy and food is really from the microbial perspective. We're getting hydrogen, we're getting methane, we're getting really yummy foods for microbes to eat on the other side. But as I mentioned here, we also have high pH. So hydroxyl ions are going to be released, and this is going to result in extreme pHs for these systems. So just because there's lots of energy in food doesn't necessarily mean that it is a great place to live. So in just basic terms, I give this talk to biologists a lot. In basic terms, we're taking rocks plus water, and we're getting life molecules, organic molecules like methane and other small chain organics. That is how life on Earth had to arise, right? Going from not life to getting life. And so that's why there's speculation that serpentinization could have had a hand in the origin of life here on Earth, where we've got just rocks and water. We start getting organic molecules and eventually could lead to life. So that has implication for life here on Earth, as well as implications for life elsewhere in the solar system. And a lot of our research was funded by NASA for this very reason. Okay, so the group that I've been working with for the past decade or so has been looking at the biogeography, so organisms kind of all across the globe of serpentinite environments. Serpentinization takes place in the mantle and in ocean crust, but when you get ocean crust abducting on top of continental crust, we get ophiolites, which means that ophiolites can be this really cool window into the ocean crust that's a little bit more excessive and less expensive to go study than trying to get a ship to go all the way out to the middle of the ocean. So here are some sites of serpentinization around the world that we've studied. The Lost City hydrothermal vent is a hydrothermal vent off of the Mid-Atlantic Ridge where serpentinization is taking place. It was discovered in 2000 And actually, Dr. Brazelton's group in, I believe it was 2018 that we went back to the Lost City and just recently got some new samples. But we've also been studying serpentinization in ophiolites as well. And from studying this kind of around the world, a model was put together. So we've got, I keep wanting to go point over here, but knowing that I need to point over here for our virtual audience. So we've got serpentinization taking place deep down here. That's producing hydrogen, methane, high pH, and those fluids are going to come up to the surface through some means. At the surface, we have oxygen. Oxygen, turns out some microbes like to breathe much like we do. And so by putting together this kind of model, we can start to look at different habitats within this serpentinite subsurface. And so by looking at serpentinization around the world, we've kind of found that in this mixing zone, so where we've got the fluids coming from deep down and mixing with the oxygen from the surface, we tend to get steadily this single type of organism called Hydrogenophaga. And it has been shown to be a hydrogen consuming organism. So it's eating the hydrogen there. But notice when we get down here where the serpentinization is taking place, we have a lot more question marks, right? We've got, are they producing hydrogen? Are they fermenting? So using sugars and turning that into energy and gases? Is sulfur important? Are they eating or producing methane? There's lots of methane here, but the truth is we don't know really what's going on down there. So to that end, we established a microbial observatory. We went out to a site on the Coast Range ophiolite in Northern California, where we knew that serpentinization was taking place, and drilled wells into the subsurface so that we could sample fluids periodically over the last decade, as well as look at rocks from that site. And so the site is up here in Lower Lake, California. It's about a 3-hour drive north of San Francisco, right through Napa and Sonoma. And so the wells that we established, as I said, we drilled into the actively serpentinizing subsurface. We have 2 sets of wells that are about a mile apart. This QV11, we're going to call this sample 2 throughout the rest of the talk, was drilled to a depth of 45 meters and has a pH of 11.5. This sample CSW11, we're going to call that sample 1 for the rest of the talk, was drilled to about 31 meters and has a pH of 12.5. So again, these are really extreme waters. For reference, pH 11, that's like ammonia. So these microbes are living in essentially ammonia. And so here you can just see what the wells look like from the surface. Here is that CSW11. We also have monitoring wells around them drilled to various depths, which actually turn out to have various different geochemical parameters too. So we can compare who we're seeing as well as some different chemicals. And here is our QV11, that's the deepest well, as well as some surrounding monitoring wells there. So not super exciting to look at the surface, but there's some really interesting stuff going on below. So as I mentioned, our first question is always, who is there? This data was generated by doing that deep sequencing of that one gene. And I've organized the samples here in order of pH. So our highest pH samples are going to be up here at the top with that sample 1. Remember, that one's pH 12.5. Going down to sample 7, which is one of those monitoring wells on the side that has pH about 7.5, so circumneutral. And this pH 2, or this sample 2 again, that pH is 11.5. So all we're looking at here, each color is a different bacterial species. And what we can see is how the species change as we go down through the samples and decreasing in pH. But what I want us to focus on are not each one of these individual species, but these groups of bacteria. So what I've highlighted here in shades of red, as well as highlighted right here in red, are what we used to call beta proteobacteria. That hydrogen Fa guy said that we always find near the surface, it's a beta proteobacteria. We also tend to find these blue guys in a lot of the wells, and these are firmicutes. That's just the group of bacteria. They tend to be anaerobic, which means they are living somewhere where there's not oxygen. So they're probably going to be at that lower side of the well where the oxygen doesn't permeate. But it gets more interesting than the fact that we see these two groups that we tend to see everywhere. This is a single species of that beta proteobacteria. And this is the percent of the total community that it's making up. This single species is making up 50% or more of these most extreme samples. That is a big deal. Usually, if we went out and grabbed some soil right now, we would see billions of different species of bacteria in the volume of soil that would fit in the tip of your finger. The fact that we see a single species making up 50% of this sample means that, not surprisingly, this is a harsh place to live, and you have to be specially adapted in order to withstand those high pHs. When we go down to sample 2, so 12.5, we see a little bit of variation. These are our field replicates, so we just kept taking samples in succession. But it's still, that single species is still making up about 25 to, let's say, 35 or so % of the sample. We're still seeing it in a high proportion around pH 10, and then it drops down to being closer to about 5 to 10% of the total community, which kind of makes a little bit more sense for a general microbial community. But, so that's the single species of that beta proteobacteria. But if we add in all the other beta proteobacteria that we saw in the sample set, what we can see is even when we get to these samples that are around pH 9 or pH 8, even though they don't have as much of that first species, they still have roughly 25 to 50% total beta proteobacteria. And so it's kind of like cousins that we're seeing down here. They're still very closely related, probably still doing similar things, but not the exact same sequence that we saw before. All right. What if we switch and we look at the anaerobic ones, those firmicutes that we think are living deeper down? Well, in our most extreme sample, they're making up, let's say, 5 to 20% of the total community. But when we get to our next sample, sample 2, you can see they're making up the 25 to 50%, which means of these most extreme samples, sample 1 and sample 2, about 80% is made up of either these firmicutes or that single species we saw before. And again, we can see that as we decrease in pH, we still see some of these firmicutes, but less and different species as shown by the different shades of blue. And in our circumneutral sample, we don't see it at all. So 2 types of bacteria we tend to see around the world in these sites, no difference here when we're looking at these various types of serpentinite samples at the Coast Range ophiolite. And then finally, just to kind of reorient us, here's the entire community. So this sample down here, the one that's circumneutral, is the most diverse. It has the most different types of bacteria in it. And we have very low diversity. Very few microbes, types of microbes are present in these more extreme samples. Okay, so that's who's there. But what about what can they do? Remember, to address what they can do, we didn't just sequence one gene, we had to sequence all of the genes in the sample. And so by sequencing all of the genes in the sample, I can look for genes that are specific to certain types of metabolism and see what their relative abundance are. How many of these genes are there present in these different samples to give us an idea of whether or not the microbes in these samples are capable of doing these types of metabolisms. Again, with our more extreme sample here, pH 12.2, we don't see any genes for methane metabolism, acetate metabolism, carbon monoxide oxidation, or sulfur. We really just see hydrogen and carbon. They're just doing the basics. When we get to our sample 2 with the 11.5, we see some more diversity in what they're capable of doing. There's some sulfur metabolism genes. There's some carbon monoxide metabolism genes. We see some acetate, and again, more hydrogen. It's not until we get to sample 3, which has a pH of 10, we still have some sulfur genes, but they're a lot lower. Some CO oxidation, but again, less than we saw in the last one. But the really interesting thing here in sample 3 is that we are finally seeing some methane metabolism genes. And then finally, sample 4, that's that circumneutral sample, kind of our control here. We don't see any methane or any acetate or sulfur. I want to note, because this will kind of come up a little bit later, the gene that we found in sample 3 is from a methane-eating bacterium. And that is relevant because there's kind of two things you can do with methane. You can make it, you'd be a methanogen, methane making, or you can eat it, be a methanotroph. Only archaea, which we'll talk about in a few minutes, are capable of being methanogens, but both archaea and bacteria can eat methane. So it was significant that this is a bacterial gene that I found there, because we looked really hard for this other type of life called archaea in these samples, and it wasn't there. We did not find them at all, despite using multiple methods. Okay, so let's bring it back to here. What have we found? Well, we still see up to 50% of our most extreme sample, we're still seeing those same hydrogen consuming autotrophic aerobes. So the beta Proteobacteria, we saw that in spades. But when we get down here, we can start taking away some of those question marks. We saw genes for producing hydrogen, we saw genes for oxidizing carbon monoxide, and we saw some genes for reducing sulfur. We did see these Clostridia, those were those firmicutes that we saw. But we also did not see, despite looking for extensively, any sign of methanogens, any sign of archaea. So we're beginning to fill in this picture. But it's important to remember that the subsurface is not this vacuum of geochemicals and water and microbes, right? There's rocks down there. And a lot of times when we're looking at subsurface microbes, we're collecting surface waters and we're inferring everything that's going on in the subsurface without thinking about the rocks, about the thing that all of the water and all of the microbes has to go through. And so that's where the next part of this talk is, looking at who is associated with the rocks and does it change the story that we have of what's going on down there. So again, we have our Microbial Observatory. We've got our 2 main wells here, our QV11, 45 meters deep, pH 11.5, and CSW11, 31 meters deep, pH 12.5. So with the Microbial Observatory, we're able to go back periodically and sample fluids. I was involved with this project until 2014, and we went back quarterly. So 4 times a year, we went and collected fluids to look at them. I'm not sure how frequently they go back now, but I think it's at least annually. We also have wells and direct access to the subsurface, so we can do downhole experiments. If we have some hypotheses of what microbes might be doing down there, we can go ahead and test it in situ underground. But what we're going to talk about now is the fact that when we drilled, instead of just drilling down and discarding what we got, we cored for rocks. And so we're going to look at who's in those. But before we get to the rocks, which I know is what you want, we need to back up a little bit, and I teased at this, and talk about life, because I realized that I might be making some assumptions about our knowledge of what life is. So this is the most current as of 5 years ago, tree of life. These are all of the organisms that are in databases, And it's looking at that one gene I told you about, that ribosomal gene. So sorry, I'm stepping off camera. We are right down here where that orange dot is. And it turns out what we usually think of as life, so microcellular or macroscopic life, falls here or here. Almost everything else on the tree of life, except for where animals and plants are essentially, tends to be microbial. So it's a very, very microbial tree. Yeah, exactly. So sorry. Online, we're at this orange dot right here, and plants are somewhere down here. But almost everything other than here and that orange star is microbial. So we've got eukaryotes. Again, that's where we are. That's generally what non-microbial people think of as life. And everything we've talked about so far has been up here in the bacterial side of the tree. What we're going to talk about next are archaea. Now, you might be wondering, what are archaea? Archaea, up until the 70s, were thought to be bacteria. And to be honest, you could find textbooks right now that don't distinguish them very well. So it's relatively new. But there are three main groups of life. We've got our bacteria, our archaea, and our eukaryotes, or multicellular organisms. And functionally, archaea look like bacteria, and they tend to be found in similar places, and they do similar geochemical things. But genetically, they're more closely related to us than they are to bacteria. And so it's important to distinguish them from each other. But we tend to find them, we usually find them in environmental samples, just like we do bacteria. Remember, I searched for them in the fluids using multiple methods, and they really just weren't there. Oh, and like I mentioned before, but just to reiterate, the only microbes that are capable, that we know of, that are capable of making methane fall within the archaea. So now we're going to look at our rocks. As I mentioned, we had 2 wells that we drilled, 2 wells that we cored. We only got rocks from the CSW11, that's sample 1, and that QV11, that's sample 2. And so here is just the lithostratigraphy that was done when those samples were taken. We've got some serpentine down here. We've got some altered mafic rocks kind of found throughout, a lot of kind of soils and detritus up near the top. And here we're just looking at some of the mineralogy of one of the deepest samples from that CSW11. With our sample 2, so that QV11, the well that goes down to 45 meters and has a pH of 11.5. Again, we're seeing some various forms of serpentine minerals. We've got some magnetite in various minerals down here. And we're also right here looking at the different minerals at the very deepest point. What I take away from this is that it's heterogeneous. It's not just like we have clear zones of what's there. And despite the fact that these are within a mile of each other, we see differences vertically. That we're not just seeing the same types of geology throughout these 2 different samples. Back to my side of things. So, as I mentioned, we took the rock samples that we got from the coring. I took them back into the lab and I smashed them up with a hammer and a mortar and pestle, and then I extracted DNA and sequenced it from all of those rocks. What we're looking at here is how much DNA I was able to get out of the rocks. You might be asking, is there DNA in rocks? And the answer is not much. It's very challenging to get out, and there's not a lot there to begin with. This line right here is the limit of detection of my meter that measures how much DNA is present. So pretty much anything on this side of the line is below detection. Anything up there, I actually have numbers for. Where we have open symbols, these are samples where we had DNA and I sent it off for sequencing and we got nothing back. It did not sequence. Anything that is filled in with color, we have sequence and data for. I feel like these open circles and things that fall below the line are actually really helpful. Sequencing centers will often tell you they need X amount of DNA to do sequencing. Well, that's not really true. I gave them essentially no DNA and they were able to sequence it fine. So just because it's below the detection or below the recommended amount doesn't mean that it won't necessarily work. And also we have some that were above detection and they weren't able to sequence. So I was looking for whether or not there was a trend here where we could say, you have this little DNA, it's not going to work anywhere else. Here, we're using a method, not sequencing, but just where we can, using some probes, just see and quantify how much bacteria is present. And so not surprisingly, up here in brown, I apologize, are soil samples. So these are just surface soils we just kind of scooped with a tube. And then here are our rocks in red. So not surprisingly, we see more cells in our soils and near the top where lithostratigraphy says that we're kind of getting detritus in soils anyway, and it drops off when we get down into the more rocky samples. Same thing over here for our sample 2. We've got our soils at the surface. We've got our limit of detection for measuring DNA or quantifying it. And our green squares are going to be ones where we actually got sequences. We see this little spike right here in our DNA, as well as in the number of bacteria in Archaea we were able to quantify down here at about 30 meters, which is intriguing to me. So, who's there? Here, we're only looking at the Archaea. And even though I started out with, I didn't count this, but let's say 20-ish samples between all of these circles filled and not, and all of these squares filled or not. I started out with about 20 samples, and of them, 5 of the rocks successfully sequenced for Archaea. The rest of them, nothing happened. But I also sequenced my soils. We won't get too deep into this. Here, class is just a level of taxonomy for bacteria. Each color is a different class. And so what we can see is our soils all look very, very similar. They all have this nitrosphaeria. This QV30, that's the one where we had this weird spike. That also looks a lot like the soils, this nitrosphaeria. And then we have different colors coming up in our other core samples, which is exciting. But because it's not just a rock in the subsurface, right? Like there was water running by it and we had to bring it up through soils to get it. I want to make sure that when I talk about who's associated with the rock, I'm not contaminating it with environmental microbes from soils or water. And so I ran some statistics to identify who is statistically a rock microbe as opposed to coming from somewhere else. And what I found when I did this is, not surprisingly, most of this QV30 sample looks identical to the soils. But the rest of our samples look pretty unique, so we can trust that those are actually rocks. I then did some more statistics to identify specifically what bacteria we can call our core enriched or rock bacteria versus soil bacteria or everywhere bacteria, or Archaea, I'm sorry. I did this for both the bacteria and Archaea, but we're looking at the Archaea here. So zeroing in on just our core enriched, so these are the ones that are statistically in the cores, not elsewhere, we can see that 25 to 41% of the total community of those different rocks were core enriched, meaning the other 80 to 60% were maybe found in soils, or statistically they couldn't really tell. So these are the ones that we're really going to focus on. But that's still really impressive. 20 to 50% of the sample are these ones that we can say conclusively belong to cores. We can see some trends, again, just some differences in color up here near the surface. So when we're closer to this like detrital serpentine soils, we see more of these blue and purple microbes. If we get down deep here, we can see more of this red and green. And then if we pop over here, the only sample that sequenced for Archaea out of all of this sample 2 was the very, very bottom one. And that one, we have some of the blue, green, and purple. I just want to note that we've got these dark blue, these red ones, kind of pink ones, and then these green. All of those are known methanogens or contain known methanogens. So we may have found our methane cycling and methane producing microbes. They just weren't in the fluids. They're associated with the rocks. As I mentioned, I did the same sort of analysis looking at the bacteria. We won't get into this in detail, but we were able to find, this is just for that sample 1, that about 60 to 90% could be attributed to rock specific, not found in either the fluids or the soils. So we have a lot here that we can work with. And then this is just for sample 2, also looking at the bacteria. This is that same sample 30 that we saw before with the Archaea. Looked like it was very similar to the soils. When I looked at it with the bacteria, it looked like it was similar to the soils too. It still had about 15% that we could call rock bacteria, but the vast majority of what we found here looked identical to the soils. But we're seeing a lot of these orange guys, these actinobacteria, and a lot of similarities between the rocks at both of or within each of the individual sites. This is very preliminary, but there is some evidence of nitrogen, sulfur, and iron cycling microbes within the bacteria that we see. There's a big asterisk there. I told you before that we needed to sequence all of the genes from the environment to know what the microbes are capable of doing. That way we have the genes that are actually involved in the eating or breathing of the certain chemicals. This, we only sequenced that one gene to know who's there. So we're kind of inferring this microbe's there, and we know from other literature that this microbe can eat something. So it's kind of like saying, well, Katrina's here, we see her there, and we know that she likes pizza, so over there she can eat pizza. I'm not great with on-the-fly analogies, but you kind of get the point, right? We're making some jumps without actually seeing the pizza-eating genes present. Okay, so let's summarize. We've got essentially 2 different samples. We have our sample 1, which was our CSW11, pH 12.2, depth 31 meters. And we have sample 2, QV11, pH 11.5, depth 45 meters. Who's there? Well, when we look at the bacteria, the fluids from both sites are really dominated, and I apologize that the colors for these flip-flopped in this figure, but they're really dominated by, here in this blue is our beta proteobacteria that we saw in the surface. It was red before. And then here in this red, and then here in this kind of brown is our firmicutes or Clostridia. Those are the anaerobes we tend to see down deep. And then red is kind of anything else that we saw. Everything else was really low abundance. It wasn't more than 1%. So it just kind of gets grouped in together as other. Similarly with our QV, remember we saw the flip-flop. We see a little bit more of those firmicutes, the anaerobes, and still a lot of the beta proteobacteria. So our fluids look very similar to each other across the 2 different wells. But with the rocks, my first observation is just, there was a lot more going on in the rocks. And I should say that for rock here, I chose the deepest rock in the data set to look at. So we just have more diversity, more microbes are in the rocks from either one of the sites. If we look at Archaea, remember there's no Archaea in the water, so nothing there, but we do see actually Archaea and diverse Archaea at that here in the rock samples. If we look at what they're doing, we see evidence for hydrogen cycling in rocks and water. We only see evidence for methane cycling in the rocks by seeing the methanogenic Archaea that we were able to detect. Nitrogen cycling, iron cycling, we don't see any evidence of that in the fluids, but we see them in rocks. Sulfur cycling, remember we were able to measure some genes for that in the sample 2 fluids, but not in the sample 1 fluids. We do see some evidence for sulfur cycling in rocks though. Carbon fixation we see in both fluids. I can't say yes or no about carbon fixation in the rocks yet because I have to dig a little bit deeper. And then CO oxidation, again, didn't see it in the most extreme fluid sample, but see it in the rocks as well as QV11. So using DNA sequencing and comparing the rocks and water, we can tell who's there in each type of site sample and what they're doing. And it shows us that just inferring what's going on in the subsurface by looking at fluids is not enough. We talked about this site right here, but I do want to say this was drilled in 2011, but there has been other drilling projects in the intervening years, both adjacent to the Lost City. We drilled in 2015 on the Atlantis Massif, and then in 2017, a group of scientists drilled in the Oman ophiolite. So even though this was the first study to start looking at this, there are more studies that are coming out right now so we can get a better handle on what's really going on in this subsurface. With that, I just want to thank my funding sources, the NASA Astrobiology Institute, Rock Powered Life, Deep Carbon Observatory, and the Joint Genome Institute. This was a very big project and we have a lot of collaborators. And I just wanted to highlight, particularly since I'm now at a primarily undergraduate institution, the students, both past and some present, who are still assisting me with this work. So thank you. I apologize if I went a little bit over, but thank you and I'll take your questions.
Thank you, Katrina. I think what we'll do, not quite sure how this is going to work. We're new ground here, but we'll start by asking if there are any questions from the audience, and maybe you could repeat the question so that the virtual crowd.
Sure.
And then if you're online and you have a question after that, I think that the technology is such that if you unmute yourself, that we should be able to hear it here. If that doesn't work, we may have to ask you to type the question in the chat. We'll start with, please just raise your hand in the chat, and I'll kind of monitor it and try to call on you, and then you can just unmute yourself and ask the question. So, are there any questions from the audience? Bill.
Thanks for a nice talk.
Thank you.
So I'm just trying to draw some generalizations from your work, and so I need a little help.
Sure.
In the sense that I work primarily near the surface, and so most of the electron transfer to sustain life is driven by organic carbon acting as electron. You mentioned that you're producing organic carbon down in the serpentinite rock area, but I'm not clear whether inorganic carbon is the source of carbon initially or not.
Yeah, you hit on actually two really important keys that I didn't highlight that make the serpentinite subsurface super challenging. So one of them is a limitation of inorganic carbon, essentially because any inorganic carbon that's present at that pH is going to become bicarbonate and precipitate out of solution, which is why, and I didn't show you any pretty pictures, if you have surface serpentinization, you're going to get travertine, and like the Lost City is a hydrothermal vent, but not your typical black smoker. It's got these big white calcium carbonate chimneys. So essentially your DIC is not available because it's tied up in rocks. But you also mentioned the electron acceptor. So as you said, we have a lot of electron donors with our organics and our hydrogen. Just like we need to eat food and then breathe out oxygen, microbes need to eat and breathe. They eat electron donors, or they eat electron donors and they breathe out to electron acceptors. So you need like a balance of those two things in order to live, to eat and breathe. There's a lot of electron donors. There's not a lot of electron acceptors. And so that's always been a question for us. Okay, well, how are they breathing? Like, where are they going with the other side of the metabolism? I don't know the answer yet. But one hypothesis we had was the rocks. Are the rocks helping them somehow with the iron minerals that are there? Could they be eating carbon or things that are around them and then breathing onto the rocks? I don't know the answer, and I'm not sure if it's known yet, but yeah, very electron acceptor and DIC limited in the system as well.
Other questions from the audience? I actually have one.
Sure.
I'm trying to, you were collecting fluids from these wells.
Mm-hmm.
Were they cased and only open at the bottom, or do you know where those fluids are actually coming from?
Great question. Yes-ish. Some of them were cased, some of them were not. Some of them collapsed after drilling. So it's a little bit convoluted. The wells that we don't have cores from, so the monitoring wells that went to different depths and had the pHs that were not as extreme, those are all cased. And we know that we just have access. We have bladder pumps at the bottom of the well. Our interesting wells, the more extreme ones, those were a wider diameter. They were not fully cased. They're partially cased, and they started crumbling in on each side. The hole started falling in after it got cased. So no, we don't know exactly where the water is coming from in those more extreme wells. And that's kind of always been a source of frustration in the system.
And one of the reasons for asking the question is that in my experience, and I'm a groundwater hydrologist, where we've seen these incredible pHs, often we've at least assumed that it was contamination from Portlandite, from the cement that was used to try to seal the casing in. And so, I mean, are you certain that it wasn't a sealant and so on that was?
Yes. I don't know specifically about a sealant, but a little bit of background about the site. So it used to be an old gold mine, and they had to drill some monitoring wells just to kind of prove they weren't, contaminating things. They then donated the site to UC Davis as a field station. And UC Davis was told, you have to, if you're gonna have a field station, you have to go out and drill drinking wells. And they're like, this water is not potable. And they were told, nope, you have to go drill. So they drilled a well, and it was full of sulfide and methane and high pH, and they kept drilling and they kept drilling. So even before it got like cased, they were like going down just trying to find something. So we have a very deep well that wasn't part of this study that just keeps going into the subsurface. So when NASA heard about these wells that were already there, that's when they got involved and started going and investigating and finding that it was really the serpentinization taking place. We did take a lot of precautions during the drilling because we knew we wanted to look at microbes. believe it or not, drillers don't really care about what they're introducing into the subsurface because you're usually just looking for your rock. And so for drilling fluids, we use sterile water, and we even put microbead tracers in there to see where any contaminants would have gone into the samples, which was really novel for this type of drilling. And the drillers thought we were insane for wanting to use sterile water, but it was an important precaution to take.
For those of us that have worked with drillers, we're not very surprised by that at all. It looks like we have a question from Margie Chan. Margie, do you want to try? Let's see if we can hear you here.
Can you hear me?
Yes, that's working well.
Okay, so my question is, if you don't have anything to sequence, just in the ancient serpentinites themselves, is there anything distinctive in terms of a biosignature that you could look at so that you could actually say in an ancient rock, even without DNA, this is evidence of past life?
Yeah, that's a super NASA question. Yes, so we actually have a collaborator, and I didn't present the data here, but who's looking at lipids. And so lipids are on the outside of cells. They tend to last longer than DNA sequences do. And so lipids are often used as biosignatures, and she has looked at those in these rocks as well. So I don't know what the biosignatures were. I haven't fully gone through that data yet, but yes, you can do that. And yes, we have done that.
So you could actually take some hard serpentinite rock and crush it and find lipids in there?
Theoretically, although I should say that the lipids that we found in here were pretty sparse. There weren't a ton of them there, but there were some. And a lot of that has to do with the fact that I think it's just really hard to live there. And I don't know about the dating of the lipids. So I don't know, that's really outside of my expertise. I'm not sure if you can then figure out how old those organisms are based on looking at their lipids. Do you know? Does anyone? It's weird to just talk into space, but.
Are there any other serpentinites that you're looking at to actually compare with these examples that you just showed us, so that you would actually know whether this is a common regular occurrence or whether you have some unusual situation?
Yeah, that is a great question. And maybe, well, it's taken me a very long time to publish this. And as I mentioned, those other drilling projects have taken place in the intervening years. And so a colleague just finished her dissertation looking at the Oman drilling. And so I was looking at that. We've been talking, and we see pretty similar organisms there. The exception being this one type of archaea that I find, these Bathyarchaeota, which are kind of really novel. We don't know a ton about them. So I have a Bathyarchaeota expert working on who they are and what they could be doing. But in general, yes, we see very similar organisms to what they see in the deep Oman samples. And that's really the only other comparable study out there. There have been a few that have done like point source rock samples from serpentinites, but nothing besides ours in Oman that has gone really that deep. We did drill into the Atlantis Massif right off of the Lost City. And there's some slight similarities, but we see a lot of differences in who is in those sub-ocean floor rocks versus the continental ones. But that doesn't really surprise me too much. Those are very different systems, even though they're both fueled by serpentinization.
Thank you.
Thank you.
Looks like we have maybe we'll take one more question from Angela Snow.
Hello, can you hear me?
Yes.
So if I understand the biochemistry correctly, it looks like the process is producing methane. So I was actually kind of surprised that you'd see so many methanogens. That doesn't seem very energetically favorable. Do you know what's going on there?
No, we do not know what's going on there. And there's a colleague of ours who's on the Oman team who does a lot of bioenergetics of these systems. And he constantly says, you shouldn't see them. They shouldn't be there. It doesn't make sense. So maybe it makes sense that we didn't find them in the fluids, but the fact that we just found them in the rocks, I don't have the answer. But I do know that thermodynamicists have been stumped by it.
Okay,
All right,
well,
thanks.
I think we will wrap it up here. Those of you that are here, I highly encourage you to at least go around the corner, because I think otherwise there's going to be a whole bunch of food that will have to go to waste or something. So please do that. And if you would join me once again in thanking Katrina for a great talk.
Well, and thank you all for being on Zoom. Maybe we can do a little happy hour once the world gets back to normal.
We do have some refreshments just to join. Is that an enthusiastic yes? Yes.